afternoon, everyone. The time is we are running short of time. Regarding research activity of IC5 with regard to quality testing materials, I'll be skipping most of the slides because already Satyam Bodole has told about the basics, and regarding seed, Vishwanath Sharma has told, and regarding nurseries, by that Sharma Bhutia will be speaking. So I'll be restricted to the what RFR has done so far only. I will go. This is already discussed, so I will not discuss all these things. So what we have done now, this Malaina arbori. Malaina arbori is Gamar, is one of the most important tree species, particularly for use for furniture. So regarding the tree improvement, we have done selection because selection is the most important criteria for the tree improvement. So we have selected these candidate plus trees across the northeast region, particularly Assam, Arunachal Pradesh, Mizoram, even for North Bengal. We have collected this list. Then we have collected the plus trees on the different criteria. So we have some colonial seed orchards were set up in the one Deoban. Deoban means this place was named as Deoban, this other campus. Naharani is in Dergaon, then Jaipur in Assam, this Agartala in Tripura, and also Imphal, Manipur. So additionally, we have collected this vegetative multiplication garden also created in our campus, Naharani, as well as seedling seed orchards were also established in Agartala and also in Manipur. So these are like seedling seed orchards, the 0.5 hectare this was established in 1999. In fall 2002, the colonial seed orchards, these are some of the areas as well as the year of establishment. The BMG basic multiplication garden, it is RFRI as well as in Naharani. These are the mass multiplication of Malaina arborea. So we have got some different clones, about 70 clones are there of this Malaina arborea. Out of the 7, we finally screened 25 clones. 20 out of these 25 clones, these are we are in Naharani area. These are there. Out of these 25 clones, 5 clones are resistant to one particular borer. This is called earlier it is Calotropis liana. Now it is called Caspatenta liana. It is major leaf defoliator of Malaina arborea. This particular these 5 clones are resistant to that particular. I cannot say resistant, is tolerant to that particular borer. We are now supplying these clones to different forest departments. Recently, we have supplied to North Bengal, the silviculture division. We have supplied. We are also doing some agroforestry model in the KVK of in Kahinkusi, then Buralixon, also in the ICR farm of Assam Agricultural University. Regarding Dipterocarpus retrosa, this is the state tree of Assam. So we have this is focus. We have selected some candidate class trees. We have got passport data for all the Dipterocarpus retrosas. Then we established the seedling seed orchard in Deoban, that is in our campus in Jaipur, also in Naharani. These are seedling seed orchards. Really long back in 1999 and 2001. Then another important Pinus cassia, this Khasi pine. Khasi pine is particularly found in Meghalaya and Manipur. So these are some of the candidate class trees we have collected from 33 locations in Meghalaya. These are the places. And also we have established some seedling seed orchard, botany trial in Uniam, in Barapani. Then Equilia malacensis is the that we have what we're doing. This established province is already certain board as well. Entire Northeast India we have divided into 16 provinces based on the gene flow barrier. We are collecting out of 16. We have found only 14 provinces. We have collected the proposals. Now we are establishing provenance resource 10 in our campus. Because we have found the different variants of physiological, morphological variants, at least 5 different variants are there. All the variances are example in our campus. And regarding the Equilia malacensis, we have been doing research in almost 15 to 16 years. For the cultivation as well as artificial inoculation. If you are interested, I can go give details about Equilia malacensis. The Melia Azadare, there is one project on All India Coordinator project on Melia dubia, but whatever the Melia, these are there, these are actually not Melia dubia, these are Melia Azadare. But within Melia Azadare, we have different variations also there. So we have come around, we selected about 296 plus trees and established 3 progeny trials. We have optimized the vegetative as and micropropagation technique of Melia Azadare. This Milia azaderic and Milia dubia main difference in the flower. In case of this Milia dubia, flower color is different. Milia azaderic flower color is lilac color. We also did one project of Magnolia Sampaka. Earlier it was called Michaelia Sampaka. So we have selected different 37 fast trees. Long back, one of this participant, Dr. Muhammad Ibrahim was the PI. We have collected these propagules from different places of Northeast India. Then two progenitors were established in Meghalaya, one in Meghalaya, another is in Assam. Then in bamboo, we have been doing this tree improvement program for bamboo for since 1998 onwards. So far, we have done 3 rounds of selection. So for the 6 species, the total number of candidate plants were about 228. So another 228 assessments were collected in next selection. So these are the bamboo improvement work we have collected from particularly Bambusa tulda, Bambusa balqua, nutans, Hamiltoni, Bambusa bamboos, and Bambusa pallida. We have collected from different places from Northeast India. So out of the 4 species, we have got 16 high-yielding genotypes. These are yield is about 18 to 26% more than the normal bamboo. These are the different genotypes. Bambusa tulda 7, Bambusa balqua 4, and Bambusa nutans 3, then Hamiltonite 2. And we are giving non-exclusive license to different these companies like MP, State Bamboo Mission, Bihar Forest Department, Deblila Biotech, our sister organization, IFGTB Coimbatore, IGKB, India Gandhi Kisi Bishabidya Raipur, and Chroma Biotech. And we have also given the license to the State Bamboo Mission. They are giving to this 9 kbks of AU. These are the ongoing programs of bamboo is going on, about 10 bamboo species. These are except, I think, yeah. Total acceptance till date is about 532. This program is for 10 years program. Then we have got on bamboo sitam. There are about 63 species are there. And then the 100 bamboo sitam is at in Aizawl, our center at Aizawl. Then we are doing the in vitro propagation of different bamboo species, this balqua, tulda, nutans, and also Hamiltoni. These are in vitro generator plantlets ready for plantation in under high-tech bamboo nursery. This is a composting yard. Then we are also giving something different, like traditionally some wild edible fruits. We are also, because under parasitic resource system, wild edible fruits, or we are having the nurseries of wild edible fruits. The sustainable impacts and future directions. What now about the quality planting material, but this quality planting material supply should be sustainable. So then a climate resilience by producing planting material that are well suited to the changing climatic conditions. So RFI contributes to the resilience of forests in face of the climate change. Biodiversity conservation, the use of genetically diverse and locally adapted planting materials supports biodiversity conservation by replicating natural ecosystem and habitats. So economic high quality planting material lead to increased timber and non-timber forest product yield, thereby fostering economic growth in rural areas. So what we are doing now, about 150 different species of forest genetic resources, we are collecting the germplasm. So we are mapping entire distribution of all these 150 species. Also, we are have the germplasm of the high yielding timbers. So we are going to supply about 50,000 seedlings to this forest department for the recent minister program also. The carbon RFS efforts in producing vigorous planting materials contribute to enhance carbon sequestration, playing a crucial role in mitigating the climate change. This is all about in nutshell about what RFI is doing in this tree improvement program. If anybody is interested in agar specially, I can give, but the time is very short. I don't know if you were interested. I can give something because they have been doing agar for so many years.
So now the session is open for interaction. If someone wants to interact with the sir.
Sir, kindly give your other. It would be nice to have a
I'll go very brief because the Equilea malacensis, earlier it was known as Equilea agallosa. The new name has been changed, it's called Equilea malacensis. So Equilea has 21 species. Out of these 21, 13 are producing agarose. So in India, I will find two types. One Equilea malacensis, this is found everywhere in entire Northeast India. And Equilea khasiana is confined to the few pockets of Meghalaya. The name is called khasiana, means Khasi Hills. Only near Mosiram area is there. Also in Hamre and in Assam also few trees are there. And you know,
More valuable.
pardon? No, because both are producing agarose. It's almost extinct. You can say Equilea khasiana almost extinct. But Equilea, whatever you find agar tree is Equilea malacensis also. But this is Equilea since 1995, 14 February, it is in Appendix 2 to regulate the trade because it is the most expensive wood in the world. So it was to regulate the trade, it is included in the Appendix 2. So now you cannot, now we can import it, process and re-export it. But recently we have done the NDF study. Now we can export it, that will come later on. So regarding the geographical entire Southeast Asian countries, you will find the Equilea malacensis from about 0 to 1,000 meter from mean sea level, it can be grown. They are about annual rainfall at 1800 3,500 millimeter, 20 to 28 degrees centigrade, relative humidity about 80%. Entire Northeast is suitable for this Equilea malacensis. Now, mostly is like habitat including rocky, sandy, more is grown based in the foothills. Now, most of our this is required acidic pH, but entire soil of the Northeast is the acidic pH, acidic. Okay. So is the distribution of all this entire different waterfront. But the base main is confined to the Northeast India, but now the other countries, this place like Western Ghat of Karnataka, because Western Ghat and Kerala climate is almost similar to Northeast India. Western Ghat, Kerala, then also South Gujarat is now they have grown. Tamil Nadu, Andhra Pradesh, Telangana, Maharashtra, Gujarat, Orissa, West Bengal, particularly North Bengal area. In Assam, everywhere the agar is there. Only this Jorhat, Golaghat, Sibsagar, Karbi Anglong, and Karimganj, here you find the natural infection of agar tree because due to the presence of one particular borer that is called Neurogena conferta. So, what the main use is from wood, you'll find chips and carvings. From chips, you find powder, oil, and incense. From oil is basically used in perfumery as a fixative, in cosmetics and pharmaceuticals also. These are main, this oil. And the white portion is full of fatty acids. It can be used for the bath soap. Now, earlier also this Maniksani was the largest buyer of this agar because it is used in the gutka industry. So these are different products. These agar handicrafts mostly produced in Bangladesh. Then agar tea, these leaves can be used as agar tea. It's very good for health. 1 kg agar tea is costing about 3,000 to 6,000 rupees. It's very popular in the Vietnam and Malaysia. That's simply like green tea. Now the youngs, the first and second leaflets are selected for making tea. There are many health benefits are there, like anti-analgesic, anti-arthritic, anti-inflammatory. Particularly, it's very good for lungs cancer. These are benefits of drinking agar tea. It's a nasal detoxifier, nasal way to remove harmful toxin. So what some of the farmers in the Namchi area, they are producing, and also in Golaghat, they are producing the agar tea. So it's a chemical composition of agar oil that I will go through. This is about agar, most of the people have seen. There is in Assamese called one Bhola Sase and Jati Sase. Main for Bhola means the tree growth is very vigorous, but no infection. Jati is normal Sase, normal infection is there. But we found that about different variants are at least 5 different morphological variants within Equilea malacensis we have noticed. Different leaf shapes are there, color is different, fruit shape is different. We have recorded at least 5 different variants. So one main problem now, agar is normally propagated by seed. The main problem is seeds are recalcitrant. So you cannot sow the seed. Immediately you have to sow. Normally from June to August, this is the period you can have to collect the seed. Immediately after sowing, you have been collecting immediately you have to sow. Otherwise, they won't germinate. So first you have to sow in the only pure sand bed. You should not use any what's called soils, FYM and all. Within 6 to 25 days, it will germinate. Within 1 month, it will germinate. So then we can transfer it to the polybags. Polybag you can keep it as soil sand and FYM. Main planting season is from May to September, April to September. This is the monsoon season. That year you can plant. So about in 1 hectare, about 2.5 to 3.5 meter. Why I am keeping this much spacing? Because now after 5th year, it will fruiting starts. Automatically it is a fall down. New seedlings are coming out. And one more thing, minimum 10 years you have to wait. Okay, gestation period. For that in intercropping, you can go for some additional crop, so they extra benefit. That's why I'm keeping this much spacing. And pitch size should be about 50 cubic centimeter. So 1 to 1.5 kg cow dung or FYM can be given, or any vermi compost can be given. The main problem one is a plant protection measure is a Hiercia bichassoides. It's called leaf-eating caterpillar. It's totally almost you can say totally defoliates the tree. Those who are having the agar tree must have experienced this one. But you should not use any synthetic insecticide, because if you use any synthetic insecticide, that will kill the main this stem borer. Better you should go for some herbal insecticide like neem kernel extract. So, some new diseases are coming up, but thing is now, in case of agar, if something disease is coming, it's very difficult to control, because this disease is caused by some fungus, because that agar is formed because of fungus only. So you should not use any fungicide. Actually, some nematodes that injures the root, through the root, fungus is make a entry. So first you have to control the nematode using some nematicide like Flurodon 3Z, then automatically the tree will survive. So under some dumping of diseases there, these are common diseases in nursery. So better you should take some either soil solarization or fumigation. Or after that, you can use some babicin. These are some systemic fungicides. Like now, because since people cannot wait for such a long time, so you have to go for something intercropping. Like here, people are using along with the agar this Phocostemon cablin, this patchouli, this sarpagandha. Then here agar along with the pineapple. In most of the apparatus, agar along with the parakanat, coconut. And most of the tea gardens nowadays they're growing agar along with that, but agar cannot be the alternative for the is not a set tree. But most of the tea gardens are using maximum pesticides, so you should not use the agar in the middle. Better you should use in the periphery. If it is organic, no problem.
That nematodes attack, you are taking that soil nematodes attack.
Yeah, there is a one particular insect called Petilencas. Sorry, nematode. It is attacking.
This attack is like at which year? Just after planting or at the when it is grown?
Not after planting. It's a bit grown.
After a few years. No?
Yeah. And moreover, why it is happening? Normally, whenever this particular caterpillar, that caterpillar totally defoliates the tree. Already plant is weak. And if the something like the water stagnates during that time, that particular nematode is come. Then you cannot use, since it is basically caused by fungus, you cannot use any fungicide. So you have to control the nematode.
The nematode is coming to nursery sleep or
Not nursery.
see, I can discuss.
In that.
There are some nematicides, organic nematicides. I can give it to you.
This particular problem was not there. Maybe only last 5 to 6 years I have come across. So we have developed some agar-based agroforestry model, because that particular farmer, it is Satyajit Bora. So we have along with the agar, he's growing this radish, broccoli, French bean, then cabbage, according to the choice of the farmer. Then like next season okra, the vendi, ginger, king chili. So he's getting some additional income, about 20 to 25,000 he has earned. Later on, we have supplied the tea and banana. So from this one of selling banana, he's earning about 50,000. The main part, my aim is so that at least he can earn some additional income till the 10th year. So these are agar along with the banana. You can grow anything like agar along this white Santala Malbama. There's a crop. The main how agar is formed. The agar is formed just basically that particular borer, that Neurozera conforta. That is the main. Once the borer attacks, fungus comes. So this borer is available only in these few places. Jorhat, Golakha, Chitlagar, and Karim. Other places you won't find this borer. Wherever the borer goes, so these are come across. We have come across 3 different types of borers. All the borers, these all are Neurozera. One is conforta, other is farina, this there. All and the other quality is different from all the borers. We are trying to rear this borer in the laboratory in artificial media. Rear some artificial media. Wherever the borer goes, the zigzag tunnel, this agar would is formed.
Lucky borer and lucky attack.
One more thing, speaking of people
The
will
same borer when it comes in an economically important plug, we call it as best.
yeah.
That
It
is
is
different, but
giving
I
something,
thought the magic is being played by this borer only.
The main problem is now with this borer is also another problem in the this Anguri Namti area. This become pest because the young seedlings are also attacked by the borer.
Then it is of no use. no?
Then it's no use. Yeah. But how to identify infected agar tree? Many people doesn't know how to identify. First of appearance of borer hole. If you see now these type of borer holes are there. If you see the grass at the base of the tree, and then you see the oozing out of water is something for the borer hole. And after about 9 months, you'll find this spindle shaped closed borer hole. Earlier, this trader they used to come and they used to see, they used to count now how many closed borer holes are there. Depending on that, they used to fix the price of the tree. So, but then these are different. Here, we have developed some keys how to identify infected tree, like appearance of borer hole, oozing out of watery substance from fresh borer hole, accumulation of frost at the base of the tree, closing of borer holes. The longitudinal cracks will be there. Leaves become smaller, yellowish in color. And if the formation of hollow sound when hammered on the trunk, then it will come. This tree is having already nasal formation is there. So, but no, but that particular borer is not everywhere. So what people are doing traditionally, they used to some traditional methods are there. They used to injure with some sharp knife or dowel. Main purpose injury is so if you make some stress condition that because of immune response that agar is producing these sesquiterpenes. Here that during the rainy season when the water deposits, that particular person agar is formed, but it will not spread. Then again, people used to put the nails, a series of nails. After 2-3 months, they used to pull out the nails. In particular person that agar is formed. Then they used to make holes and they are putting the molasses. Main purpose not if molasses they will attract this fungus. These are different methods. Now these are bamboo sticks. Bamboo sticks are immersed in some chemicals. They are putting. This is the mostly in Thailand. They are doing it. In also this Karnataka, they are also doing this method. Like this is called CA kit method. They have this one University of Minnesota. They have patent. They are using chemicals like sodium chloride, jasmonic acid, salicylic acid. But main thing is if you use chemicals, the tree will die after some times. But the quality of the agar is also not that good. So this in China, this is a very huge tree. They are making some the wounding in the tree. Even the hot iron ore they used to put. So we have been working here for almost like 15 years. So we have found that almost 3 to 4 different fungi are there. These are responsible for agar formation. These fungi are with us. We are multiplying these fungi in laboratory. So we are making holes depending on the height of the tree. About when the tree is about 8 to 10 years old, about 35 to 40 centimeter girth, we are making hole, leaving the 1 meter from base. We are putting this with the help of syringe. This one. Then after, because we can cut the tree, and a particular person we used to remove the bark, we used to see the difference of the color difference there. They become brown to black. So this after 2 years after inoculation, entire tree is getting infected. This one, this whole now, this is spreading this infection. This is in Meghalaya. We have been doing it in the 5 districts of Meghalaya. This is in Tripura. Tripura Forest Department given one project. Entire 8 districts of Tripura we have done it. All the trees got infected. Now they are extracting oil. This is in Tripura. They are after 8 months itself they are extracting oil from this one. This is Kailas or in Tripura. This is in West Bengal, particularly North Bengal area. This Rajabhat, Khoa, and Lataguri of North Bengal. This is West Bengal Forest Department given up this project. This is Dumduma. This earlier one. This additional PCCF, I think this Akashdeep Borua is yes, we are comparing it with the traditional method as well as our method, and we are extracting the oil from the traditional method. We are what the composition of the oil. We are almost similar quality. We are getting it. This is in South Garo Hills, Meghalaya. This is in Tamil Nadu, in Dibrugarh. This is in Nepal. Last year, we on the request of the Nepal Forest Department. I went to the Damoli in Nepal. We have trained the forest department also in that Tanao district in Nepal. This is in, okay. Some of the trainings we have given in the Meghalaya. These are also Meghalaya. This is a West Bengal Forest Department. They are doing one, the Sangeeta Dubey. Now I think additional PCCF, she was also there. This is North Bengal. Then from 2021, 19th August, award this product, now this Sasi inoculant, Honorable CSCM has released for commercial purpose. Since then, we have been selling it. So we are selling this paste about 600 rupees. One packet is enough for 2 trees. And this broth, we are selling it for 480 rupees. This is about trade and all, that I will go to details. This the agar is you can say that's a goal. About presently it is from 500 to 12,000 rupees per tola. Just like gold is called is sold in tola. 1 tola is 11.66 gram. These are agar root chips. But the main problem is agar. This is now different case, but there is no scientific grading. Based on the more the black color, more the price. That's why people are sitting the people, because they are mixing something like coal tar, making it black. There is no agar. So it should be, but there should be some GCMS analysis about the resin content. Then only you can say that it is a pure agar. So they are removing the white portion with the help of Botali. So this is this agar chips for agar oil. No, they used to put in the immersion water for 14 days. Then these are different method of water distillation. Critical method. These are traditional boiler. It's called deck. Mostly find in the Namti, Amguri area and Naharani area. These are pure agar oil. These are about 115 to 16 lakhs, you can say, per liter. Then mostly for agar ships, it has gone to this UAE, the Dubai area, and for oil is going to this France for perfume. These are the mostly among the trade of agar product, mainly Saudi Arabia, and followed by Singapore and Kuwait. Among the exporters, Singapore, Thailand, UAE, Indonesia, Malaysia, top positions. These are all about. Then Assam Agar Promotion Policy here, that among the details. These are Assam Promotion Policy. Incentives are there, different incentives, international trade. So there's a recently, not recently, I mean, yes, January 10th, new SOP has come up for agar. That is details is there. Director General Foreign Trade, they have given the SOP for agar, how to sell it. The procedure is here. So, yeah. So we have since RFI is the scientific authority of CITES. So we have done the NDF. Then we have fixed the quota. About 25 tons of agar can be agar root chips can be exported, and 1.5 ton of oil can be exported. This is about this one total planter, and in Sibsagar, Saraida, Jorhat, these are Manipur and Tripura. These are all economics of the agar production per hectare. We have observed that about you can total expenditure 1 hectare will be 31 lakhs, but main expenditure will be inoculation. We have fixed about 300 rupees per tree for inoculation. That way come about 4,70,000. So if the trees are young, below 10 years, if you have to give under booster dose, so under 4,76,000.
What is the spacing?
Yeah, that is 2.5 to 3.5 meter in the year. Yeah, that way 1,700 plant. But we see return, this is estimate only. About gross return is about 2 crore 35 lakhs. But that I can't say this estimate only. But whatever we have done in the inoculation, up to maximum up to 1 lakh, they are selling per tree. If the trader this is giving the 1 lakh to farmers, but that means trader is selling it more than at least 5 lakhs. So wherever happened, particularly in this North Gar Hills, Kharkutta area, the people are very poor. All the houses are kapsa houses. Now after selling the agar trees, inoculated agar trees, now all the houses are kapsa. But I don't know how they're legally or how they're doing it, but this trade. But whatever we have done, everyone 100% success is there. Now, okay, this is all about in short in this agar. So if any query, you can just.
Can this trade be done legally, like export of oil?
No, that is there. You can do it.
I have been meeting various farmers, and these people are dependent.
No, already SOP is there. given by the Director of Foreign Trade. Recently, this agroforestry board, Agar is also included. So definitely can be legal.
We are also saying that it is there.
Yes.
And I am also propagating to an extent the agro plantation. But I'm saying that when I met with farmers, they are saying that it would no perfect rate nine.
Yeah, that is there. Yes, yes. Yes, yes. Yeah, there's so many. Yeah, there's so many things are there. That the bigger agar grade is a bit totally subjective. So that's why people are taking advantage of these things. This one. So there,
It's a kind of mystery.
yeah. So that is the main.
The house can overcome all these problems of the farmer.
Yeah.
Go a long way because
Yes.
at the ground level there are a lot of issues.
But that's why we are now giving agar seedlings to many tea gardens. Because now they are.
But if they don't use chemicals because they're using lot.
Because now most of the they converted to the organic tea. There are empty spaces in the tea gardens. Recently, whatever the empty space there is 5% or 10% land should be used for the this other.
Protecting shade tree has become so difficult, you're talking about.
No, I'm not talking. This agar is not a shade tree.
What I'm saying is.
Yeah.
We are finding it so difficult protecting our shade tree from villages, from our own laborers, because firewood is a requirement.
Yeah.
Imagine if you plant shade agar, which is worth 1 lakh rupee a plant. How will we protect it?
Vandalism is also a big problem. Elephants,
Yeah.
elephant, cattle,
Yeah.
and two-legged.
It is possible only for small tree growers.
Yeah,
Because,
yeah.
you know, literally, small tree growers have their houses within that area. So they can.
But these tree you own eaten by cattle. Cattle means particularly cow we don't own it. Only goat can eat, but cow doesn't. Not easy. Goat. Cow doesn't.
So it's feasible in all the district of Assam.
Yeah, yes. But only nasal infection is not there.
In your presentation, we could see now only few districts.
No. They are nasal infection because of the presence of the particular borer. Other trees, the agar trees are there, but there is no infection at all. You have to depend on artificial inoculation.
So as per modern method of RFI, they say that if the tree which we plant, the agar trees, there has to be inoculated. Inoculated, the result will.
No, that's not. Particularly, the borer is available only in few districts. That too, borer is very, very selective. You cannot forcefully take this borer. It is very, very selective.
আপোনালোকে suppose এটা plantation model দিছে যে
হয়।
আগৰ এটা facilitate কৰিছে
হয়।
এইটো মডেলৰ কাৰণে আপুনি যে কৈছে যিটো ইনফেকশন হ'ব বৰাৰ ইনফেকশনৰ কাৰণে তো আমরা ফাঙ্গাস অ্যাটাক হ'ব এইটো তাৰমানে আৰ্টিফিশিয়ালি আমি কৰিব লাগিব
কৰিব লাগিব, yes.
এতিয়া বেছি সাকসেস
হয়।
For your technology, artificial innovation technology of RFI is more advanced because then uniform infection and elective infection caused by nasal.
Difference in the quality.
No, quality is not different because that we have one of our students. He is doing PhD. She has analyzed the oil quality for both natural as well as our artificial. All the almost components are same components we're getting. And one more thing that because since ours is natural fungi, only putting it artificially. What we did after inoculation in Tripura earlier, because the agartala means beneath the agar tree, but now there is no nasal infection in Tripura. But after artificial inoculation in Tripura, trees in Tripura, now nasal incidence of that particular borer is increasing. Okay, thank you.
Thank you so much, sir, for your presentation. May I now invite the next invited speaker for today's event, Dr. Panipan sir, who is currently serving as MPhD and the Group Coordinator Research of ICFI, that is Eric Research Institute, Jodhpur. So he shall be delivering his lecture on recent advances in policy planting material. To inform all of you, sir is an expert in plant tissue culture. He has been working on forestry tree species in the arid and semi-arid zone. So over to you, sir.
Thanks a lot. I hope I'm audible. First of all, a big thanks to Director RFRI for inviting me to present and to be part of this great workshop, which is a technical workshop on quality planting materials. And as you can see, I come from Arid Forest Research Institute, and in some ways you can see the Rain Forest Research Institute, rain and arid, these are antonyms. They are, if you have a scale of agroclimatic zones, then I work in altogether it's 180-degree other side of the story. And here it's rains, and there we have drought and all those things. But I mean, saying that and saying that about the differences, there are a lot of commonalities. And it is because of those commonalities that I'm here today and talking about this particular aspect. And since my area of research is molecular genetics and also biotechnology, so we have been trying different ways of how we can, the aim is finally how we can improve upon the planting stock, that is the planting stock improvement programs. So to begin with, let me just have a very brief background, because how diverse things are. We all know about our country. It has got its very diverse ecosystems. The forest cover wise, we are 10th largest globally, though it's only 21.7% as of the recent report. But we have 91,000 documented animal species, 45,500 plant species. So India is very rich in biodiversity. And not just that, out of the 36 biodiversity hotspots, we have got 4 biodiversity hotspots right in our country. So that makes India very promising in terms of biodiversity. But all is not that good. Because when we talk of our forest, when we talk of our plantations, particularly the natural ones, the forest, I mean, the biggest problem is that we have one of the poorest productivity of the plants that we have. The productivity is one of the lowest. And that is the biggest challenge. Low productivity of, we may have high biodiversity, but productivity is low. Now I personally feel there are 3 challenges that we are faced with. The first one is land degradation. So apparently here in Assam and in most of the part of the country, we don't come across that much of land degradation the way we see it from AFRI, because we are working on the problems of marginal lands, the problem of salinity, the problem of mine overburdens. So many things are there. But when we talk of land degradation, degradation of the terrestrial ecosystem, that has actually been primarily because of the human intervention. We human beings, as we all know, are a greedy species. We always think of ourselves. Even as Sir was telling about agarwood, there is the plant has a problem. The plant is infected, but we are happy. We want it to be infected. So we are that greedy. So because of that greed, we have destroyed our lot of land which was good. And degradation of terrestrial ecosystem. I mean, I come from a place, an institute which is on the fringe of desert. Now, desert ecosystem in itself is very good. I mean, there is nothing wrong with desert. It's an ecosystem. It has got its own plants, its own flora, its own fauna, and we need to preserve that. We need not convert the desert into a green belt. That is not an intention. Intention is we have to stop it from spreading further. The desert should not spread further, and degradation should be stopped. And you'll be surprised to know that it is if you take the global picture, globally the entire Earth, the complete land surface, if we think of 25% of the land is degraded land globally. But in India, it is 39.5%. Almost 40% of the India's land comes under degraded land condition. So it's not just the arid and semi-arid region. There may be degraded lands in Assam as well. So and that has caused a decline in a lot of biodiversity, and that leads to decline in ecosystem functions and services also. The cost of this degradation is in terms of Indian perspective, it is over 31.27 billion rupees is the cost of land degradation. And as you all know, in 2012, there was the Sustainability Conference of the United Nations, which is also known as the Rio Plus 20. And a document came out which said the future we want, the future we want. And that future we want talks about land degradation neutrality. Can we neutralize the land that has been degraded? And that is known as LDN. So we have to go back and we have to reclaim our lands which have been degraded. Now, you may be wondering why I'm talking about this when we are here to talk about agroforestry or talk about quality planting material. But all this is connected. Because until we have technologies for land degradation, we cannot think of productivity increase and enhancement. Everything is connected because it's a vicious circle. And the degraded land reclamation, when we talk of, the best thing is today we have to increase the trees outside forest. And to increase the tree outside forest, agroforestry is considered as one of the key ways of reclaiming many of our forest lands. Now, the challenge number two, and of course, I forgot that in India, in COP14 in 2019, our Honorable Prime Minister Shri Narendra Modi ji has declared that 26 million hectares of land in India has to be brought into the fold of LDN. That is, we have to reclaim this much of 26 million hectares of land has to be reclaimed. And this is in terms of India. India has got 130 million hectares of land which is degraded. It's only 20% land that we have to degrade, and it looks such a mammoth task. Now, talking to our challenge number two, that is low productivity. So degradation, we have on one place we have degraded lands, and the extent of degradation is somehow increasing over as time is going on. As we are cutting more and more forests, as we are doing whatever we are doing, we are degrading the lands much more. Degradation is not just because of deforestation; it is also because many things like industrial effluents, they are released into the land. Land becomes of no use. There is salinity, secondary salinity. So many things we are doing. Our industrial revolution has caused a lot of degradation, and we have at the same time the forest productivity, which is very low. Indian forests are one of the poorest in terms of forest productivity. You'll be surprised to hear about the figures. The productivity is only 0.7 cubic meters per hectare annually, as against the world average of 2.1. So we are much below the world average. The global average is 2.1 million cubic hectares, and we are just 0.7. So inherently, genetically, our forests, the productivity is low. And that is one of the biggest problems when it comes to quality planting materials.
What do you mean by low?
Low means the productivity is low. Means that when we talk of productivity, productivity is in terms of seed production. It is in terms of growth rates. It is in terms of the way they can fight infections. It's like it's a gamut of things that define the productivity. Productivity means at a physiological level, it is the photosynthetic rate. It is also in terms of carbon sequestration rates. So overall, the productivity is quite low for us, and also not just that, the average growing stock is only 32 cubic meters per hectare as compared to the world average of 110 cubic. So this is also, growing stock. The way the amount of growth that takes place is also much less in Indian forest. Now it varies from region to region. We have the rainforest here in Assam; they may have high productivity, but putting together the entire country, the situation is not very good. So we are sitting on a resource which is the productivity is low. And the third problem is the complication of the modern times is the climate change. We all know climate change is something is not at all helping us anyways. We have land degradation, we have low productivity, and on top of that we have climate change. And 1.5 degrees Celsius rise by 250 is going to be catastrophic. There is an estimate that even 1 million species may face extinction because of this. And last year in India particularly, and in west and north, we have seen tremendous heat wave. The temperatures were 51. Incidentally, I'm also the NPC of National NEEM Genetic Improvement Project. And under this NEEM project, you will be surprised to know that we could hardly collect neem seeds because flowering did not set in because of heat wave. All the flowers were there, good flowering was there, but flowers just dried up and withered away. And so as a result, the seed set was very low. And neem is very important plant. You know, neem is important for neem oil, azadirachtin, neem coated urea is required. And all the northern and western belt, we had very poor neem collections. So that was one of the big problems. And let me tell you, because of climate change, the chances of having such heat waves increases 51 times more. If there is a climate change, that has made these kind of episodes happening 51% times more, 100 times more, in fact. So India has done a lot of things to, there have been policies. The biggest role that was supposed to be played was by the forest development corporations. They have done massively well, but still have been problems. Because when we talk of the forest development corporations, they are supposed to do aggressive forestry for production. It's basically production forestry. But the problem is that many of you come from representing the industry, the timber industry. And the Rajasthan, Jodhpur particularly, is the hub of timber, the handicraft industry. The biggest handicraft industry comes from Jodhpur. Now, the biggest problem is that FDCs are producing less than 5% of the timbers. The timber that is coming is hardly coming from FDCs because there are problems. Because they are sitting on low productivity over forest. The mother plants, from wherever the seeds are being procured and being produced as propagules, they are basically having low productivity. So the problem is everything is connected. And therefore, FDCs have also fallen short of their objectives. And we are nowhere close to becoming a wood producer, country which can even complete its own demand. All the wood is coming, a lot of wood is coming from Germany, from now China, and many other countries. The wood is coming, and we are not able to meet up our own requirements. So that way, not fully, but yes, agroforestry has a ray of hope, not from the timber point of view, but from the point of view of land reclamation, and at the same time also for some subset for industry also. And that's why agroforestry is being considered as one of the major mainstays for wood production in India. That is why agroforestry, government is pushing agroforestry to that extent that we have to think of agroforestry. Now, different institutes, all the ICFR institutes are working on those lines. Can we make agroforestry lucrative to the farmers? Because farmers are also, first of all, farmers won't allow trees because that takes away their land, which could be used for agriculture. So, as you all know, SMAF has the logo of per made per paid. I think that is a very well-known fact. So that is the kind of drive that is required for increasing the forest cover outside the known forest. That is the RFAs. That is the recorded forest areas. So recorded forest areas can't be touched. Plantation forestry through FDCs has limitations. So agroforestry becomes an important aspect which has to be looked into. The solutions are, of course, to have quality planting material. We have been discussing the whole day. We have been talking about QPMs, but then there are caveats. The afforestation programs, plantation forestry, agroforestry, all depend on quality planting material. There are set SOPs, there are set protocols for producing quality planting material. But the prime reason why we are not taking off, we are not moving in a big way is, as I said, productivity of our forest. That is where the selections we are doing. That is where the lack happens. Until we are improving the quality, we cannot move ahead. And quality comes from long-term tree improvement programs. So finally, it comes down to long-term tree improvement programs. India went into the tree programs, the long-term tree genetic improvement programs started way back in times of when Britishers were here. That is when they started doing it. But for almost close to a century, things were moving at a very slow pace. The reasons were that there was lack of commitment, lack of funding. So the genetic improvement program, because we are talking of unlike agriculture, here we have plants, trees which have long gestation period, and it takes very long time to be able to produce a clone or a variety of a species which is genetically superior or genetically improved variety. That is one key issue. So long-term tree improvement programs is something which is very important. But right now we are in 21st century, and there is biotechnology. We also have to embrace biotechnology to aid us in moving in a fast track manner to genetically improve, produce genetically such trees which have desired traits, high productivity. So I call it actually not just QPM, I call it Q square PM. Q square PM because we just don't talk about quality. Quality is suffering because of low productivity, but we are also not getting high quality in high quantities. Quantity is the biggest problem here. Quality is lacking, but quantity is even a bigger problem. So it is Q square PM, quality and quantity planting material that we require. So quality will come through long-term planting stock improvement projects. And it was when ICFRE was established for the first time in a very concerted manner that ICFRE and the institutes, the 9 institutes of ICFRE, RFRI, AFRI, TFRI, all different institutes, sister institutions, we started working in a big way. There was a planting stock improvement project program of the council that kick-started with funding from the World Bank. And World Bank funding initiated some of the programs which actually are now at a place where ICFRE is able to say that yes, in the last 2 decades or 3 decades, we have got something that in the coming years we will have some species for which we can have real high quality planting material with real genetic gain. Because the point comes here is when productivity is low, we have to increase the genetic gain. We have to talk in terms of genetic gain. Until we talk in terms of genetic gain, we may be doing lots, but at the end of the day, productivity is low, economics will be low, everything is well connected. So, and then now it is the age of biotechnology, gene mining, gene cloning, gene transfer. This is the norm of the day. And the government of India has also, in 2-3 years back, they released a white paper by the NITI Aayog, in which also shows that yes, they want to embrace this genetic engineering pathway. The government of India wants to take this genetic engineering pathway, and in fact, genetic engineering has been even genetically modified rubber has been outplanted in the field. Field plantation of genetically modified rubber has been done in India. Cotton is a success story for India. The BT cotton is already, whatever shirts we are wearing, it's all 99% genetically modified cotton. But there is a lot of friction when we talk of genetic modification. It's a new field, lot needs to be done. And when we talk of quantity, if we have good quality, we have to increase the quantity. And quantity can come through vegetative multiplication, that is macro propagation protocols, and also micro propagation, that is tissue culture based protocols. So I'll take you quickly to through some of the works that we have done in terms of improvement of the quality and also quantity. I'll talk about two particular species. They may not be very much relevant to your region, but this is just showcasing the technologies there in the country. And let me also share one more statistics with you that I was talking about ICFRE, that they have done a lot of work on genetic improvement. In fact, ICFRE so far has released around 70 varieties of around 17 species, out of which 11 are tree species. So ICFRE and we have new programs are starting now. ICFRE is working in an all India coordinated program mode. So we are in fact trying to do in a fast track mode already. I'll just share with you some of the case studies. Case study one that we have done that I have also been involved in respects. This is the case of neem. I think neem is also one of the species which grows here in Assam also and many other places. The only problem is neem doesn't come up in cold climate. Wherever there is temperature goes down below 2 degrees or even 4 degrees for a extended period, where there is a frost attack, neem doesn't come up. But there are right now 11 agroclimatic zones out of 50 in India where neem grows. So as way back as in 1992, the National Provenance Trial of neem was established, and it had 39 seed sources from all over the country. And this was a mega program that was started, and it was in 92, and already we are in 2023. So many years have passed by, and still we don't have much out of this work, because this work is now coming towards a point where we can give some results. But still, so much time has gone by because we are dealing with tree species. But at that time, when initially they started working, they only worked on height and girth as traits, not as oil and ESA. And then they had an international provenance trial which came up in 1995 under the FAO, and this was several trials were established in Jodhpur, Jaipur, Palanpur, Jabalpur, and Coimbatore with 18 provenances from different parts of the world. And they primarily worked on just girth and height class to just find out the superior germplasm with where the growth rate is high. And at present, these trials are only continuing in Jodhpur, Jaipur, and Coimbatore. Then there were progeny trials. Now, naturally, as I won't go into the details of this classical approach, but I'll just tell you that in Gujarat, massive scale under board a project was there, and they selected 367 CPTs from Gujarat, and they did oil and azadirachtin estimations, because azadirachtin is the key secondary metabolite for which neem is used as a for its known qualities for the insecticidal properties. So they found that neem, there is a wide variation in ESA and oil, and they did just 1 year data, and 1 year data was not enough. But still, they were able to, select 17 CPTs, and this work paved way to a second project by IFCO, which was received by FRI Dehradun. And FRI Dehradun, after again their own research on ESA and oil, they were able to release 6 high-yielding varieties of neem in 2021. Now, AFRI has also taken up work on, AFRI has been working since the beginning on neem, and different aspects have been worked out. And one of the key aspects was also, because when we are talking of high yielding varieties, we have to clone, we have to preserve that quality. We cannot go through seedlings, because if we go through seedlings because of segregation, we will lose the character. So we have developed tissue culture based protocols for neem, and this is a well reproducible protocol and high efficiency protocol for neem tissue culture. And these were, even hardening was done, and the hardened plants were raised in the field. And this technology was transferred to the Gujarat State Forest Department, and they have, they are using our technology to do tissue culture for multiplication of the neem trees. Then recently, as part of the All India Coordinated Project funded by Campa, we are also doing more work. In India, we have selected 1,000 CPTs from different parts of the country. 6 institutions of the council are involved in this, and we are in fact trying to. The main reason we are doing this project is because in our last project, we found that 1 year data is not enough, because there is so much of variation in azadirachtin and oil content because of the climatic variations. We thought, let us see pan India, which are the 2 best agroclimatic zones for neem, so that we can have finally the best quality planting material for neem. Neem is a plant which is required right now so much for production of neem coated urea and also liquid urea. And neem oil is, if we want to go for eco-friendly pesticide, then neem oil is one of the best. So this project aims at finding top 50 CPTs we want to select in this, and we want to identify 2 best agroclimatic zones where the selection should be made, where the productivity is the highest. Because usually, if we randomly select from anywhere, we may end up with plants where giving us nothing. So this is a scientific endeavor where we are just trying to look into all those aspects, and not this. These are some of the, as you can see from some of our selections, some of the CPTs. We are documenting even the quality of seed, the variation in seed, and we are also trying to do oil extractions in house and trying to test the oils in ESA. And these are some of the results of our oil in ESA from across the country. For the first time, this kind of work is being done at such a massive level. And not just that, we have also used biotechnology for genetically modifying, that is, introducing a foreign gene into neem. And under this project, what we are trying to do is we are using Agrobacterium tumefaciens, and with that, the main aim is first to develop a protocol for gene transfer, which we have successfully already developed. As you can see here, this is the protocol for gene transfer to neem. Our aim under this AICRP 26 of neem is that we want to genetically improve neem for cold endurance. Because as I said, neem cannot grow in colder regions. It doesn't grow at beyond a particular altitude. If we are successful, we hope the aim is that one day we may have neem which can have, we make it cold enduring. It can even be planted at higher altitudes at colder regions so that we can produce more neem seedlings, more neem seeds for oil extraction and as a bio pesticide producing plant. So that is the prime reason. And so this is a neem work. It is under process, and we have got some good results. And I hope in the years to come we'll have something for the country to have better neem varieties. Now, so this is all about quality planting material of neem. But I now come to a second aspect. That is a second case study, which is about a plant called a small tree called Google. It's Google. You might have heard of Google. Google is called Commiphora vitae, and it's a plant native to the Indian desert. But it is endangered. It is critically endangered. Actually, it has been so badly overexploited for the oleogum resin. It's also a Divya in Ayurvedic system of medicine. It has been so badly overexploited that it is hardly available in some patches in India now. It is on the verge of extinction. So we took up this challenge of first doing an assessment of how the populations are. We tried to go to all the pockets, all the 33 divisions of the districts of Rajasthan were surveyed, and then we did under a coordinated project of funded by NMPB, and we we collected accessions from everywhere and established a germplasm bank at Afri. Then we also developed molecular markers to study, because molecular markers are a tool to study the population structure, the variability, the level of variability. We are talking of a species here, Commiphora vitae, which is so much overexploited that the genetic base is very narrow. Until we have a wide genetic base, a species cannot survive. We have to have a wide genetic base. Here, we have developed molecular markers also for quality control, because we also found that in this particular plant, there is another problem. Besides overexploitation, the natural regeneration of this plant is extremely poor. Only 5% natural regeneration is there. Moreover, male plants are virtually non-existent. So it is basically apomixis, apomictic pathway it is following. And this plant is just vanishing from the face of earth. So we developed tissue culture protocols for this plant. And when we do tissue culture, we also have to know whether we are cloning or not, whether we are making true to type plants or not. So that's where also this molecular markup system is helping us to find out, to check the genetic fidelity, the genetic reproducibility of our plants. And then we have also worked very hard on very extended period of work we have carried out on the hardening procedure of this plant, because tissue culture, you can produce tissue culture. Tissue culture protocols can be fairly easily made. If some plants are difficult to do tissue culture with, some are very easy. But the ones which are easy also, they fail, because at the end of the day, you are not able to plant them in the field. When you do outplanting, the plants don't survive. So we worked hard on this particular aspect, that is hardening of tissue culture raised plants. And we have established several field trials, and we have got 100% survival rate in our field trials. And not just that, we also work, this is another aspect actually, we have also worked on setting up a bioreactor for production of its Google gum, that is Google Steron. It produces Google Sterons. So we thought, can we have bioreactor setup? And in fact, we have developed a solid state bioreactor at Afri, for which we have also filed a patent. Let's see what happens with that patent. But I mean, just in case, just to alleviate the pressure that so that natural population is not touched by the people, the tribals who are extracting the gum out of it, so that the plant is saved. That was the ideology. These are just three protocols that we have developed. First protocol is this one, where we start with cotyledonary node, and this the rate of multiplication is not very high in this. This is a seed dependent one, so it is not a very good protocol, but this was our first protocol. Then we developed a second protocol, and this was from, that is micropropagation from the shoots. And this produces the clones. This is one of the good protocols that we have developed. And this is our third and the best protocol, most highly efficient protocol, where we have actually achieved cyclic somatic embryogenesis. We just have to start with material, tissue culture material just once. Once we have the cultures, we can go on and on and on for years together by producing plants through somatic embryogenesis. And somatic embryogenesis mimics zygotic embryos. That it just produces like a seed. A small plantlet is produced, and it just, if you give it proper conditions, it will germinate with a root system and a shoot system. You don't have to provide it any rooting hormone or anything. Without hormones, it will germinate. So this is the best protocol that we have. And we have not just that, people also say that tissue culture is expensive. So we also worked on understanding the economics of tissue culture. We found that our somatic embryo protocol gives a plant which is only 19 rupees per plant, and the cotyledonary node and axillary bud based protocol produces plant which is 27 rupees. Because you can see the somatic embryo, this third protocol is so efficient, the rates of plant produced is also low in terms of this. And we have done DNA marker study on this. Everything works perfectly fine here. And I'm almost coming to the end of my presentation, but I'll just also share with you that AFRI has also released high-yielding clone of Shisham recently after work of 2 decades. That is, after work of 20 plus years on Dilbirja Sisu. This was some other group which was working, and they have in fact produced this 3 clones. ICFRE and Ministry has finally accepted these clones after DS1, 2, and 4. And this is with a 25-year rotation period of Shisham. It is expected to produce wood at 284 cubic meters per hectare and merchantable log of 84 cubic meters per hectare, with a total income of 29.40 lakhs is estimated from the merchantable logs. So this is a technology that is, I mean, just to tell you, it is slow paced, but yes, we are moving in the right direction.
Will it adapt to the
Pardon?
Will it adapt to the
Yeah, actually, this was a regional variety release. So we will be testing, because we have got, in fact, these are just 3 clones that have been released. We have got many more, around 34 clonal material is there. We have to test it. So once we have done this, we will now in the second phase going for the country level testing of these clones. So once that is done, we'll come to know whether how good they are for Assam. But particularly these are the clones which are suitable for primarily the regions of Gujarat and upland Rajasthan. So no, not really dry. These are for slightly more rainfall oriented regions. They will not survive in the regions where it's too dry. This is in the regions which are subhumid, moderately. So they might be able to do, but I don't know. I mean, I don't know. We cannot say at this point, because we need to test. Until we test, we cannot say. Yeah, but I mean, see, this is just one example. ICFRE has released so many varieties, 69, 70, I think, 70 now, 70 varieties of different species. And many of them will be tested in a national setup. Once that is done, so there will be availability of maybe this variety, or maybe this particular clone or some other related clone may be suitable for this region also. So we'll come out in the years to come. I think we'll have some good material, because this genetic gain for which we have been working for so long, 2 decades we have been working, now the benefits are, the rewards are being reaped now. And then just my last slide on a way forward about the Q square PM, as I call it, quality and quantity of planting material. The first thing that I feel is we have to ensure the long-term planting stock improvement programs. We have to have species-specific programs. There are a lot of programs that are going on in the country. There are some programs which were initiated, but midway they have stopped. So we should not start again. We should start from where they ended, because we have got data We should revive those projects which were going on in the past, and we should start from there. Long-term projects should be there. There should be continuity over periods, because what happens is, say I am working on a particular plant, I retire, and that work ends there. It should not end like that. The person should be appointed. These are policy decisions. A person should be appointed for working on a particular species. There should be species specific specialists in the system. Then we should ensure proper funding continuity for such kind of missions like the moon mission we have. And we have to go in mission mode, and we have to also ascertain quality certification things. That is something very, very important. Until we are able to give us quality certification, nobody is going to listen to us. Nobody will believe us, yes, that this is going to, this quality certification has to be integrated into these programs. Then the second thing I feel is we should ensure quantity, as I said, quantity through large scale tissue culture. It's time we should blaming tissue culture, saying tissue culture is too expensive. We should embrace it as a technology with benefits for quality, for quantity production of quality materials. And we should ensure people participation in tissue culture. Tissue culture is not just now anymore a lab-oriented high-tech thing. It is a technology which should go to the masses. The technology should transfer. People should come up with tissue culture labs. Already this is happening in Rajasthan. We have got several labs. In Gujarat, there are tissue culture labs. And there is hydroponics and tissue culture both coming up, being taken up by the industry. Industry should come out for setting up funds for this. And these researches should be funded by industry also. Industry should also fund research in this direction. Then we have to have robust distribution networks and the supply chain, which for which industry itself has got its own expertise and logistics. So if this is integrated with research, I think we can really work wonders. So industry and research has to come together in a big way. Only then, otherwise you will fall short in getting the material, will fall short in supplying. Until there is a fusion, there is a marriage of the two. And then the other thing that I would say is it's time we should focus on genetic improvement, fast tracking it using genetic engineering tools. Genetic engineering is also now a thing of the past. We are now talking of genome editing. Genome editing is like not introducing a gene, but editing the genome, because genome is made up of A, T, G, C, its alphabet, the four alphabets of which our whole genetic material is made up of. It's time now, already technologies are there, even ICFRE institutes are working on editing the genome, and that we can change by not making much. And Government of India recently, DBT has I should say, they have in fact deregularized. There are three types of genome editing. The SDN, one type of side-directed nuclease, one type of genetic engineering, genome editing has been deregularized. There is no requirement of biosafety framework to get involved and stop it from doing. So that framework is now coming up in the times to come. I think we'll see more of genetically modified things which are acceptable by the masses. But yes, there is a caveat. We have to be careful when we do talk of gene transfer things. We should test them thoroughly that they are not going to damage our environment. We are into forestry, we should be sure about that. So, and last but not the least, certifications, as I said, I would just reiterate once again, quality control protocols using DNA markers, that is our DNA barcoding and DNA markers should be there, and there should be incentive programs and incentives, and there should be subsidies and tax exemptions should be given to such things. Only then the price, because these technologies, biotechnologies are very, very expensive. Sequencing, DNA markers, all make the certification becomes very, because the cost of certification is very, very high. You have to bring down certification by providing subsidies, tax exemption. Those things have to be there. So policy, there's a lot of policy matter involved in all this. We have to think about that. So that is all. I hope it was a little too technical, but I hope it was not that boring. Thanks a lot. Thank you, sir. Thank you. If there are any questions, I would love to answer them.
The session is open for discussion. If anyone wants to come forward their views. So if there are no further questions, I thank Dr. Tarun Khan sir for sharing his views regarding the importance of genetic improvement and plant tissue culture in ensuring the availability of 2 square PMs. Thank you so much, sir. May I now request our respective director, sir, to kindly welcome Dr. Bhavesh Gogoi, soil scientist from Assam Agriculture University, Jorhat. I again request, sir, to kindly felicitate our invited speaker, Dr. A. Babu, Director of Fertility Research Institute, with a memento. I request Director sir to kindly felicitate Dr. Tarun Shah sir with a memento. We shall now proceed with the fourth session, that is on quality planting materials, nursery and nutrient management. So for the first lecture of this session, we are now invite Dr. Karma Bhutia, who is currently serving as scientist in this particular institute. He holds his specialization in forestry and more specifically in medicine. They are now invite Dr. Karma to start his presentation.
Thank you, Dr. Karmakar. Respected Director, sir, ICFRA, RFRI, respected Director, T Research, and all the esteemed participants, and my senior colleagues and my dear colleagues. So, I'll be talking on the basic concept of nursery techniques and how these nursery techniques can be helpful in producing quality planting material. So now let me start with the introduction. So as we all know that forests plays a significant role in maintaining ecological balance besides providing various benefits to us due to indiscriminate and over-harvesting of extraction of timber, fuel wood and other forest produce. Our forests are facing tremendous pressure. Although various initiatives like social forestry, community forestry, national forest policy, national agroforestry policy, etc., emphasizes massive plantation efforts, particularly in the non-forest areas. So, in order to achieve the national forest policy's target, that is 33% of the forest cover against the current forest cover, that is 24.62%, agroforestry holds a great potential and can contribute significantly in increasing the forest cover of the country. So the major constraint faced by all these programs or the initiatives has been the lack of availability of the quality planting material. So this issue can be addressed by raising the quality planting material through proper nursery techniques. So here the nursery plays a very crucial role in producing as well as supplying the quality planting materials. So now coming to the what is nursery and why there is need for nursery. So nursery is simply an area where the plants are raised or grown for eventual planting out. So it consists of nursery beds, paths, irrigation channels, etc. So coming to why there is need for nursery. So particularly in case of addressing the problems like those species which do not seed every year, like inconsistent seed availability and having short viability or the low germination percent, and those slow growing species, here we can go for a collection of seeds, and then we can make it available through nursery. And then in case of roadside avenue plantation, where we require tall seedlings and sturdy seedlings, and which can be achieved through nursery techniques. So in many times the direct sowing doesn't give us reliable results. So in this case, we have to go through planting of the nursery raised seedlings. And similarly, the casualties replacement can be done by the by planting nursery raised seedlings. So if you go by sowing the seeds, then there's maximum chance that it will be suppressed by the weeds. So the planting of nursery stocks is the best method to go for introduction of exotics like poplar, eucalyptus, and it is also very economical because uh, in nursery in small areas we can intensively manage all these planting and other operations, and this nursery plays a significant role as a tool for addressing the issues like reforestation, restoration, and conservation needs. So now coming to the basics like the types of nurseries. So there are different types of nurseries based on different factors like the duration of their use and their irrigation facilities like planting stocks. In case of their use, we have temporary nursery and the permanent nursery. And this temporary nursery is we maintain it for a very short period of time, uh, just to provide the nursery stock for the plantation sites. So it is normally smaller in size and suitable for mainly the hilly regions. And in case of permanent nursery, so these nurseries are maintained for the long period of time in order to provide the sustainable supply of the nursery stocks. And this is normally usually uh, established in order to meet the demands of one or more forest ranges. So generally it is larger in size, and on the basis of irrigation facilities, we have dry nursery where the nurseries are maintained without irrigation or artificial watering. So it is mainly dependent on the rainwater. And in case of wet nursery, we maintain it with irrigation and artificial watering, especially during the dry seasons. And on the basis of planting stock, we have naked root nursery and the container nursery. So in case of naked root nurseries, so we plant these plants are grown directly in the nursery soil, whereas uh, through cane or any yeah. So in case of container nurseries, the plants are grown in first container, and along with it's grown in the growing media. And now coming to the types, the nursery beds. So these nursery beds are specially prepared portion of land, especially for raising the seedlings in the nursery. So there are different types of nursery beds on the basis of various factors like vertical level with respect to the ground. So there we have two types, that is raised bed and sunken bed, as you can see in the pictures. So in case of raised bed, so this is usually suitable for high rainfall area or the moist area. So here we prepare it by raising the bed around 50 centimeter above the ground level with the support of either you can use bamboo or bricks or sometimes stones. And normally the standard size in forestry is 12.5 to 1.2, and it depends on the location and the topography. And in case of hilly region, it can go up to 1.8 to 1.2. And like species like the amla and the Buchanania lungeon can be grown by raised bed. And in case of sunken bed, it is usually prepared in the dry areas. And normally the sunken beds are prepared by digging 10-15 centimeter deeper than the normal ground level. So this is suitable for those species which has hard stick coat like the Terminalia species. And on the basis of the plants growing in them, we have seedling beds and the transplant beds. So seedling beds are those beds where the either seedlings are raised or the transplanting in other beds or for planting up. And in case of transplant beds, so we either seedlings are raised in the seedling beds or they transplanted before planting out on the site. So now coming to the nursery site selection. So this is it's very important in context of the quality planting material. So there are various factors that we have to consider while selecting a nursery site. So selecting a proper nursery site plays a very crucial role in producing a quality planting material. So the first, the location. So the location is very, very important where we establish nursery. So the location, that it should be centrally located for the uh, located. It should be located near to the plantation sites as far as possible for the easy transportation of seedlings. And there are many other factors like it should not have like permanent shade, and it should be free of some disease and the insect infestation. And in case of the other factor is water supply, so the nursery site should have permanent water source. And and then coming to the soil, so in case of soil, so it should have well drained sandy loam soil, and the pH should be between 5.5 to 7.5, and it should have some moderate fertility and some organic matter in it. And then coming to the other factors aspect, which is also very, very important. So in case of Western Himalayas, uh, we can go for selecting the northern aspect. And but in the higher altitudes, the southern aspect is preferred because it becomes very cold. And in case of Eastern Himalayas, only up to 1200 meters, the northern aspects is preferred. But beyond that, westerly or the southwesterly aspect is preferred. And now coming to the slope. So the nearly flat or the terrain with good drainage is usually preferred. But in case of the hilly areas, so slopes up to 5 degrees can be considered. And coming to the labor availability, this is also a very important factor when we talk about the nursery site selection, because raising nursery is a very labor intensive work. So as far as possible, the nursery site should be located near to the area where there is the availability of the labor. So similarly, the land availability. So the adequate land should be required for producing the seedlings, and there should be some additional space available. So in case if there is more demand in future, so for the purpose of expansion. So now coming to the nursery design and layout. So this is also a very important factor. So this depends on the what kind of plant or the species that we're going to raise, and then the site topography. So there are various factors that has to be taken care of, like nursery while laying out the nursery design and the nursery layout. So we should have there should be well defined and the protected boundaries by using fencing. Like we can go for, like in case of permanent nursery, we can go for barbed wire fencing, and in case of like some temporary nursery, we can go for some live hedges and all. So there should be proper roads and inspection paths, and then facilities like storeroom space, labor huts, garages should be made available, and there should be platform for the purpose of seed weathering and for the seed treatment, and the facilities for seed germination area, and for the central growing area where we'll be growing the seedlings. And there should be space, additional space for the future expansion. And the important one is irrigation facilities. There should be permanent irrigation facilities, and it should be provided with water tanks or maybe sprinkler system. And there should be proper drainage system, which is very, very important. And seed beds, like facilities like seed beds, mist chamber, shade house, working area should be properly made available. And there should be a soil storage shelter where we can go for storing the soil or the potting mixtures or etc. And then there should be space for compost making. Now, this is the simple nursery layout. So the layout of nursery is very, very important if we consider, if we as far as the uh, as far as the quality of planting materials is concerned. So nursery, usually the nursery layout, the shape of nursery is in rectangular in shape, so that it can be divided into different smaller rectangular blocks, leaving space for various other facilities. Like as you can see in this picture here, we have like different establishment like facilities like office, storerooms, vehicle shed, labor shed, and mist chamber, shade net house, drying shed, compost area and the seedling beds that you can see and the water tank. So there should be a proper path inside and the road for the easy transportation, and it should be connected with the main road, as you can see in the picture. So now coming to the containers and its type. So containers, we use it for raising plants from the seed or for the transplanting purpose. And now coming to there are different types of container that are being used in the nursery, but like poly bags, root trainers, Jiffy pellets, dona, baskets, tubes, earthen pots. So among all these different types, so poly bags, which is being widely being used because it is cheap and easily available and in different sizes. So and then we have root trainers, which is also being widely used. So this main purpose, as you can see in the picture, the main purpose of using root trainer is to prevent the boiling of roots, and this allows the ear pruning as the root comes out from the there is opening from the bottom of this root trainer. So as the root comes out from the hole, then it gets dried out and then gets ear pruned, and then it allows for the development of the fibrous root system. So and coming to the Jiffy pellets, so you can see, as you can see in the picture, so these are made from the compressed peat, and this is a very soft material and which can easily be penetrated by the roots, and it can be planted along with the plants. So similarly, we have dona, which is made from leaves of Bauhinia vellae and etc. And then we have basket like prepared from bamboo strips and all, and we have tubes which can be prepared from autumn tiles, bamboos, paper, etc. And we have this autumn pots, which is, it is relatively costly because because of their weight, it increases the cost of transportation as well. So now coming to the infrastructure and facilities. So the infrastructure and facilities like shade net house, glass houses, poly house, mist chamber, poly tunnels, propagation boxes, growing chamber, compost unit, office cum store, and water source. So these are very, very essential when you talk about raising the quality planting material. So now coming to the one by one, the first one is shade net, as you can see in the picture. So this shade net house is prepared by using mostly the agro nets. So this is mainly made for the purpose of providing shade to the young plants, especially those shade-bearing plants, and to protect them from direct sunlight and the excessive temperature. So usually 50 to 70% shade is provided, and then sometimes even the bamboo strips and then the thatches are being used for this purpose. And then coming to the glass house, and the glass house is mainly made from the glass, and this is the main purpose of the glass house is to provide the control temperature, the conditions like temperature, control temperature, irrigation, humidity, light condition, for the better development and growth of the plants. And it is comparatively costly as compared to poly house. And now coming to the poly house, so this is poly house is made up of polythene sheets. So here in the poly house, it maintains higher temperature, especially during the winter seasons. So it promotes the optimum plant growth. So this is mainly widely used for the propagation of off-season seedlings, which face high price in the market. And now coming to the mist chamber. So this mist chamber is similar with the kind of the poly house, but it is supported with the mist installation. So it is mainly used for the initiation of rootings of cuttings. So where we maintain high relative humidity, that is above 80%, and so it is mainly used for the hardwood cuttings. And now coming to the polytunnels, as you can see in the picture, this is made up of plastic sheets forming a tunnel. So this polytunnel mimics the greenhouse effects. So it helps in providing the protection to the seedlings from like the adverse weather condition like cold, wind, rain, storm, etc. So because of the greenhouse effect, so this improved in the germination and the plant growth. So now coming to the propagation, so we have the propagation box for this chamber. So this is also mainly used for the propagating techniques. So, but especially for the small operations, like for the lesser quantity of plants raising or for the smaller plants, and this chamber or the propagation box provides high relative humidity. And now coming to the compost pit. So the nursery also must have proper compost unit in order to supply the sufficient quantity of compost. So this is mainly made in the pits. So the usual size is 2 meter long and 1 meter wide and 1 meter depth. So it consists of 3 different layers. Like the ground layer will be soil, then comes to leaves and grasses, and then the manure. So here, except for the ground layer that is soil, so each layer that the grass and the manure is water, and then they are compressed by working on them on top. It is done continuously until the depth of the pit is filled up. So now next important infrastructure facilities, office come store. So it's very important that nursery should have a proper office come store. So in order to keep records of all the nursery activities and the registered notebooks, information books, all this, and even for storing tools or the raw materials and the manures, fertilizers, etc. So now coming to the very important aspects of nursery techniques, that is the nursery operations. So there are various operations that is being carried out in nursery. So it is very essential that all these nursery operations should be carried out in a proper manner. So the first coming to the preparation of soil working, like various nursery operations includes preparation of soil or the soil working, seed or planting materials quantity determination, sowing time and the method, potting mixture, irrigation, shading, weeding, transplanting, maintenance of soil fertility, nursery register, updation, protection against diseases and insect pests, and the hardening. So now coming to the first point, that is preparation of soil and soil working houses. So this is very important as far as the quality planting concerned, because there are various steps involved in the preparation of this soil working. So deep plowing should be carried out in order to provide better irrigation, and then the plantation sites should be free of like stumps, stones, roots, and any other undesirable materials. And the ideal soil medium is considered a mixture of loam and the sand. And in case of the prevention against the white ants, which is the pest, so we can go for application of Termex, benzene, hexachloride, Aldrex, etc. So, and in case of the soil management of the soil pH, so in case of the acidity, we can go for the application of lime. And in case of the countering the high pH, and we can go for the application of ammonium sulfate. And now the next one is coming to the seed or the planting material. How can we go for a quantification of it? So this planting or the seed, planting material or the seed is quantified in terms of the weight. So we can, by using this formula, as you can see here, the W is equal to A into D, the bracket closed by P into N into 100. So where here we need to have the idea of the area of bed, the size of the bed, and the number of plants required per bed, and the plants percent, and the number of seeds per gram. So accordingly, we can calculate the requirement of seeds in the weight. So usually in practice, the quantity of seed required is twice for the drilling sowing method, and almost 4 times in case of broadcasting sowing. So now coming to the sowing time and the method. So this sowing time and the method depends on various factors like the seed ripening time, species growth rate of the species, and the size of plants required for planting out. So usually in case of the majority of most of the species, the spring sowing is commonly preferred or the practice, and then it depends on the seed viability and the germination, and those seeds with low viability and the seed must be sown immediately, otherwise there will be drastic decline in the germination. And whereas in case of the long viability seed, it should be sown, especially particularly during the low temperature or low or the moderate temperature. So the season between like the months September to October or February to March. And there are different sowing methods involved in the nursery, like we have line sowing, divulged into small pits or the dribbling or broadcast. So this method of sowing also depends on the various factors like seed size, germination percent, and the type of container that is being used. And the normal sowing depth is usually twice the diameter of the seed. So now coming to the potting mixture. So this is also one of the important aspects of the nursery practice for raising the polyplanting material. So this potting mixture, the better the potting mixture, the better will be the health of the quality of the planting material. So good potting media is essential for the development of healthy and the fibrous root system. So what are the qualities that potting media should have? So it should be light in weight, and so that you'll be convenient in transportation, and it should be well drained, and it should have good water holding capacity, and it should provide good aeration, and it should not have high salt content, and the potting mixture should be free from the weed seeds, nematodes, and diseases, and it should have slightly acidic soil with good cation action capacity. And normally the sandy loamy soil is the best choice for the nursery preparation. And as a thumb rule, the soil should be the growing medium. So for different types of soil, like for heavy soil, like clay soils, so we can go for mixtures like soil, fine gravel, and the organic matter in the ratio of 1:2:2. And in case of loamy soil, we can go for 1:1:1. And in case of sandy soil, we can go for 1:0:1. So potting mixture can be of either inorganic or the organic type. So inorganic like sand, gravels, vermiculite, perlite, etc. And organic component like peat, charcoal, compost, other waste material, etc. So now coming to the other aspect is the irrigation, so which is very important for the plant growth and the development. And so the irrigation has to be properly done. So in order to raise the quality planting material. So the optimal nursery watering time is usually early morning or the late afternoon when there is the low temperature, so that the evaporation rate is less. So and then there's better utilization of the water by the plants. And there are different methods of irrigating it, and we can go by using cane, atomizer, percolation, and sprinkler, and the flooding. So similarly, we have shading, which is also very important, particularly in species like Magnolia champaka, which is the shade-bearing species. So to provide shade when it is very young, and also provide protection to the frost and species in frost prone areas, especially in the morning. So this shade also provide protection from rain, the adverse condition like rain, hail, and other adverse weather conditions. So the other important operation is weeding, which is also very important. So as we all know that weeds compete with the seedlings for moisture, nutrient, light, and space. So there must be regular removal of the weeds in order to prevent the suppression of the seedlings. And this weeding becomes very crucial, especially when the seedlings are very young. So there's it can easily be suppressed by seedlings can easily be suppressed by the weeds when it is young. So coming to the transplanting. So transplanting simply means the moving the seedlings from the seed beds to the other part of the nursery or into the polypods. This is mainly done for the purpose of preventing the long taproot, and allows the seedlings to develop a fibrous root system. So, and it is also done for the in order to achieve and appropriate the optimal size of the seedlings for subsequent planting out. And then the another important operation is the maintenance of soil fertility. Although this topic will be discussing in detail by the next speaker, and So like normally this fertilizers, it varies, the fertilizer requirements varies with the species to species. So usually 20 tons per hectare well decomposed farmyard provides good essential nutrients, and the recommended doses for certain species like eucalyptus, cheer pine, and teak uh, uh, given as you can see here. And now the next the one is nursery register, which is very, very important. And so this for the purpose, or you can say nursery journal, for the purpose of keeping record of all the activities that is being done in the nursery, like seed collection, time sowing and transplanting, uh, seed supply, and all the nursery operations, and should be maintained in order to properly monitor the activities of the nursery or the operations, nursery operations. And the other important factor is protection against the disease and the insect pests. So we talked about the quality planting material. So when we talk about quality planting material, so one of the major factor, the important aspect is the it should be free from pests and diseases. So nursery, the seedlings, the plants are usually infested with various insect pests like worms, termites, white drops, Epiphyllida, etc., and diseases like damping off, weed, root rot, powder mildew, etc. So there are several management practices available, like chemical, biological, culture methods for the management of these insect and pest and diseases. So among all these the different methods, mostly and mainly the chemical methods are being commonly practiced. So the next factor is like aspect is hardening. So which is very, very important. After when we grow when we achieve the seedling then we go for hardening. So hardening is very, very important operation. So in order to survive for the seedlings, especially in the harsh condition, the environment, outside environment, when we take it to the field. So this hardening is done in order to acclimatize the seedlings to the outplanting shot. So this is usually done by reducing the shade and then exposing the seedlings to the direct sunlight, and also sometimes reducing the fertilizers and the nutrients input. So this hardening it's very helpful in minimizing the seedling mortality in the field. So this is the diagram which is showing the various steps that is involved in raising nursery through seeds. Raising nursery planting stocks through seeds. So as you can see here, so this has been developed by the ICFRE. So as you can see here, we have taken three different types of seeds. That's very minute seeds with good germination. For example, eucalyptus and kadam and spruce, and then seed with erratic or poor germination like teak, amla, gamar, and the other one is medium or the big size, big seed with good germinations like sesame, neem, babul. So in case of this very minute seed, we can directly go for the sowing in the germination beds. So it doesn't require pre-sowing treatments. Whereas in case of seed with erratic or the poor germination, we should go for pre-sowing treatment, as you can see in the picture, and then it should be followed by the sowing in the germination bed. Whereas in case of the medium or the big size seed, yes, sure. I'm almost done. So subsequently, we go for, once we are done with the pre-sowing, then we go for sowing in the either in the poly bags or the root trainer. Then subsequently, we go for transplanting it, and then after that we go for hardening process. Then ultimately, when the plants are hardened, then we go for planting out. And similar in the case of the cuttings. So here we can see the cuttings are different types. Like we can take different species have different potential for the rooting. Like very difficult to root species like eucalyptus, and the moderately difficult to root species like ficus, and the very easy to root species like poplar willow. So in this case, especially very difficult to root plants, we go for coppicing. So we take the cuttings from the coppice shoots, and whereas in case of moderate and very difficult, we go for direct cutting from the branch cutting. Stem cutting, and then we go for treatment of the cuttings by using like the rooting hormones like IBA, IA and INA, and then we go for subsequently planting in the rooting medium and inside the mist chamber in order to provide the congenial the condition for the root growth. Then subsequently we go for the rooting cuttings, and then we are subsequently we plant it in the root trainers, either in the root trainers or the polybags, and then we expose the plants to the direct sunlight and reduce the shading, and then this is the hardening process. And once this is progressively done, and then once the plants are hardened, then we go for the planting out. So this is the example that I'm showing it as an example. This has been developed by the CAFRI and the ICRAF. So here they have published the nursery, the standards planting stocks. So where they have come up with all the important around 40 plus important agroforestry species. So I've given only 10 here. So like in case of poplar, you can see the age. That is when it is 12 months, the height should be 300 to 400 centimeters. That is 30 to 40 feet. And in collar diameter, that is 3 to 4. So similarly, for the other species, they have given the standards. So similarly, we need to go for, we need to follow this kind of standards. So they have given for that in terms of the quality assessment. So coming to the conclusion, so what are the challenges and the way forward? So at present, various plantation and the orchards mainly depend on the unregulated private nurseries for their planting material. And then due to the various growing demand for the nursery species, so most of these nurseries which do not follow the recommended nursery practices for producing the quality planting material. So coupled with the lack of proper guidelines or the training in the nursery industries also results in the production of low quality planting material. So therefore, there is the need to regulate these nurseries to enable them to produce high quality planting material using scientific interventions. Thank you.
So thank you, Dr. Diallo, for your wonderful presentations. Now the session is open for discussion. If anyone wants to put forward their questions to this other speaker, may do so. So if there are no further questions, then we can move forward to the next speaker. Dr. Bhavesh Gogoi, who is a soil scientist working in Assam Agriculture University, Jorhat, and we shall be providing this lecture on nutrient management practice for QPM.
Good afternoon to all of you. So myself, Dr. Bhavesh Gogoi. Actually, I worked here earlier in this institute for 10 years. After that, I joined the Assam Agriculture University. So I'm a soil scientist. Actually, I'm interested in this cropping system and farming system research. Also, I'm interested in this agroforestry research. Presently, I'm engaged in an all India coordinated research project, integrated farming system. So the topic allotted to me for delivering this nutrient management practices for QPM. So I'll take not much time. Within 15 minutes, I'll be completing. So QPM, the importance of QPM, quality planting material, all we know. So here I'll be talking about only means why this nutrient management is important, as well as what we can do, means how we can go. Means here I'll be talking mainly regarding this means some organic mode of this organic sources of nutrient. So this we know that this plant, that is 17 essential nutrients are there. Nowadays it is 18. Cobalt is included, and in case of means animals also will be required. It's 18 essential nutrients are there, but the nickel is not required. Instead of nickel, iron is required. And in case of any human being, we know that we are requiring 19. So all will be requiring this level being will be requiring these nutrients, and these are the essential nutrients which are required for plants, means growth and development of the plants. So these are some of the deficiency symptoms, means if soil will be lacking of these essential nutrients, so the plant will suffer from these nutrient deficiencies. So you can see this first one, this is the nitrogen deficiency, V7, typical V7, this deficiency will be there. And second one, this is nothing but this phosphorus deficiency and this potash deficiency accordingly. So this often we confuse with this nutrient deficiency along with this water stress and chemical injury and some disease attack. So these are some of the micronutrient deficiency in case of the agricultural crops. I'm showing this, just wanted to show you how important these nutrients are for growth and development of the plant. Means if it is less, then it will show some deficiency symptom, and if it will high, then it will lead to some toxicity symptoms. So like that, you know, if we'll be applying more and more amount of this fertilizer or the chemicals fertilizer to or nutrient management, if it is wrong, then it will lead to the pollutions. Means this first picture, that is blue baby syndrome, symptom, that is due to high amount of, if you will be using high amount of nitrogenous fertilizer. And in our drinking water, if the nitrogen concentration will go below 10 ppm nitrogen concentration, then this blue baby symptom will be there. This is fluorosis. And this last picture, that is this gray baby symptom due to cadmium deficiency. Sorry, cadmium toxicity. So these will be there. This is eutrophication. You all know sudden algal bloom. This is due to mainly nitrogen and phosphorus. It will be, these are all due to this unscrupulous use of these fertilizers and all. And again, this if will be using more amount of nitrogen fertilizer, that will lead to the global warming. Means this will be releasing more amount of this greenhouse gases like this methane, nitrous oxide, all these things will be there, and this carbon dioxide will be releasing to the atmosphere. So if we see the nutrient status in India, so most of soils are this in case of so far as nitrogen is concerned, the soils are very deficient. Besides that, this phosphorus is also, the soils are deficient. So this deficiency is due to this crop removal, leaching, volatilization loss. Lots of factors are there. We know that this average soil loss from in our India, it is 16 lakh, I mean, it's 16 ton. This is the average soil loss rate in our Indian soil. So this, if you can see the deficiency nutrients, this the nitrogen is the most deficient nutrient in case of our Indian soil, followed by this among the major nutrients in case of this phosphorus, potash, and sulfur like that. And among the micronutrient, this zinc is the most deficient nutrient in case of Indian soil. So likewise in the year 1950, the nitrogen was only the deficient nutrient. It was identified. But later it is going, and in 2000 we have found a lot many nutrients it's become deficient in case of Indian soil. It is mainly because of this more amount of mining, imbalance nutrient management, just because of this. So that is why this balanced nutrient is very important. So plants require balanced nutrients for growth and development of the plant, and we know that this nutrient mainly means nitrogen. This is required for growth and development. Phosphorus, that is required for root development, and potash, that is required for disease resistant or for getting a robust growth, means seedling or something like that. So that is why this balanced nutrition is very important for quality planting material production. Because if we will not be giving these nutrients properly, then we cannot expect this quality planting material. Because like that of this saying, the morning source the day, if the seedling will not be good, if seedling root system will not be good, so definitely we will not going to get the good growth of the plant later on. So fertilizer, we know these are nothing but some chemicals. So these different types of fertilizers are there. This can be used if we are selecting some class 3, say. So for that class 3, we can go for, we will have to go for management, fertilizer management to that crop. So like that of this, some methods of fertilizer application is very important. We are applying the fertilizer. Means if we are managing some plantations for, say we have some guava plantations. We will be taking some layering or some layering, and also we'll be taking some grafting, we just say some cuttings from that plant. So at that time, we'll have to manage that regular plantations with some fertilizers and some, some organic manures and all. So for that purposes, we need to know the different types of methods of applications. Means in case of this, means 4R, means light, rate, right time, right source, right method. This is very important. In case of plant management also, or in case of any plantation management also, for getting the quality planting stock, this fertilizer management is very important. So these are different types of methods of application, we can say, of fertilizers. So this, as I said, that the source, rate, and time of time, this is very important. And besides that, this native soil fertility is also very important. If the native soil fertility is good, then that means fertilizer requirement will be very, very less. That is why the soil testing is very, very important. Say in case of this native soil fertilizer, this if the native soil fertility is good, it means nutrient supplying capacity of that particular soil is good, then fertilizer requirement will be less. And if the native soil fertility will be bad or it is inferior, then we'll have to apply more amount of fertilizer. So right source, right time, right place method, if we'll be applying these chemicals or chemical fertilizers or in an integrated mode, then there will not be pollution and all the entire environment. So these are different types of organic sources of nutrients for the plants. So these are, we know this FYM, the compost, nitrile, all these will be requiring for this maybe for preparation of the protein mixture and all. These are all these raw materials from where we can prepare the compost. These all raw materials can be converted to well. So this is the availability of this bio waste, we can say, in northern region of India. We can so all these raw materials we can convert it into, means say this vermicompost or liquid manure, or we can use this bio waste or this biogas production. And what about the biogas slurry that will be coming out? So that can be used in the nursery or for this tree management, or for, in any mixture also we can use that one. It is not like that we'll be using 1:1:1 this sand, soil, and this FYM. We can use the vermicompost also. We can use enriched compost also. And how we can prepare this enriched compost and vermicompost, we can I'll show you. And we have used that one, and we are getting a very good result out of that. So you can see this one. This is steps involving composting, and this is Windows methods of composting. So these are the aerobic composting method using Raxco and Azolla. We can use this one. We can prepare this type of compost, and this compost can be used very easily for preparation of this composting, means potting mixture, or means what do you call it, this mixture media for sowing the seeds as well as for planting the seedlings. So this is how this can be enriched. The first one, this living compost materials we can take, and after that we can add rock phosphate also. 10% rock phosphate we can add there, and we can also add this compost wash, and then we can add biofertilizer also. 1% biofertilizer is sufficient, and we can mix this and we can prepare this enriched compost. And this enriched compost we can use for this preparation of this compost mixture. This will be giving very good result. So this was used in this Fujolokia production in our university, and we are getting good result out of that. This mixture was this soil as well as this compost, enriched compost. 1:1. This we have used, and we are getting a very good result. And this you can see, these are the planting materials we have produced from that type of particular means media. Here we are using 1% soil, 1% sand, and 1% enriched compost. And we are preparing this what do you call it? This citrus, lemon seedlings, as well as this papaya, as well as the oregano. And we are getting a very good out of this, good growth of these crops. This papaya we have planted there in our integrated farming system model, and we are getting a very good growth of these crops. And these seedlings we are distributing to the farmers also for some from our private projects are there, the TSP projects are there. So out of in that project we are distributing those seedlings. So this vermicompost also we can use in those media. And vermicompost, when we'll be talking about this vermi technology, it is not like that only the vermicompost production will be there. So this will include this vermicompost as well as this vermiworm and this vermiwash. This Bharmi wash can be used for, it can be extracted for the various ways. We can say means these are low cost vermicompost production technology. We can use this Bharmi wash, and this Bharmi wash also we can use this prepare this and this compost also. This can be extracted. We can take 1:1% means 1% this compost as well as 1% water. We can keep it for 2 days or 3 days, and after that we can extract it out this wash. And this wash can be, this can be used for spraying the crops also. This seedling in nursery you can spray, and this liquid manure. In our integrated farming system, we are preparing this liquid manure. Means whatever this leftover urine as well as this manures are there. So leftover material that used to go to that chamber. We used to have this liquid manure tank. Or liquid manure tank means chamber used to be there in that tank. Step by step from one tank to another tank it will go, and later on it will, from the last tank we used to collect those liquid liquid solution, and we used to spray to the seedlings. And even in the agricultural crops as well as in the vegetables also we can spray that one. It will not means like their foliar spray. It will not give to the food to the plants. Besides that, it will repel the insect also. It will spray this one. Disease and pest will also be less. So we are getting these results. Very good result out of this. So this liquid manure, this can be sprayed. Even this is a very good. There is a very good scope of marketing of this one also. This liquid manure. It can be applied like this way also. Some farmers used to apply. So we have even oil cake also. We should not forget regarding this oil cake. This can be used also for this media preparation. Even we are nowadays we are using this our turmeric mound also for preparation of the media. So this turmeric mound also it is rich in calcium as well as the secondary nutrients and all. So this can also be used, and we are doing lots of things. Means we are doing some research also on that. So this oil cake is very rich in nutrients, and this can be used. And this biofertilizer that can be mixed for this and this compost preparations. And these are having lots of advantages, no need to explain, I think so. So these are different types of biofertilizers. This biofertilizer, these are the benefits. This can increase the yield from 10 to 40%. And even it will help in the growth and development of the plants. And even this can reduce the use of these chemical fertilizers, because this can supply, say, if you will be using rhizobium in case of legumes, then this can add 25 to 30 kg nitrogen to the soils. So after growing these legumes or any sort of this leguminous from that particular soil can also be used for preparation of this. I mean to say this potting mixture, because fertilizer, we know this biofertilizer application, there are 3 methods. We can go for seed treatments, we can go for root treatments, or we can go for soil application also. It means we will be growing this leguminous from in a particular area, in a particular soil. That soil later on, after harvest of this legume crop, we can use for preparation of the potting mixture, because that will be very rich in nutrient. I mean to say, more particularly in case of the nitrogen as well as the phosphorus. So these are green manure crops. I mean to say, this green manure crop also play a very important role in case of in case of nursery or in case of quality planting materials production, because you see they used to add the organic matter as well as they enrich this soil with nitrogen and also with some microbial means population used to increase like anything. So these are different types of green leaf. From this green manure plant, we can use the green leaf. This is the way by which we can add this into the fields. And these are different types of green manure plants. The leaf, they are very rich in NPK, so this can be used. And these are the here in our stations, we are growing these green manure crops. After that, we are producing very good quality planting materials of coconut, and we are selling this one. And out of this 150 and 200 rupees per this planting unit per seedling. So like that, we can grow this green manure crops, and after that, we will have to plow it down for 45 days or 40 days, and this soil will become very rich in biomass as well as nutrient content. And even that, if we can use that soil also for preparation of this potting mixture, so that can give a very good result, I think. So here we are getting these very good results. Means these coconut seedlings we are producing. Not only coconut seedling, as my background is agriculture, so I am giving this example. So you can use this any sort of this means material. I mean to say this can be used for other tea species also. This method. So last I want to say that this whatever soil we will be using, that this analysis is very important. We'll have to means analyze the soil for N, P, K as well as for nutrients. Means and after that we'll have to grade them. Means whether it is medium in nutrient content or whether it is low content or high content over. So accordingly we'll have to mix this nutrient. I mean to say we'll have to mix this equine or fertilizer or biofertilizer or whatever it is in here. Means any fertilizer is there. So in that way we can prepare a very good potting mixture for going or for means raising the seedlings from the seeds and all. So last I want to tell that this soil is very important because in a healthy soil only we will be getting this good growth of the crops as well as good growth of the trees, and it will give the good means food and good human beings, and in a good human being also you can get the good means I mean to say there will be good fish or so we can use the soil, but we should not abuse the soil. So this is what I want to say. Thank you for giving me this opportunity. Actually, my health was not good when sir gave me this invitation for giving me this lecture. From next day itself, I was suffering from illness, so I did not get any time for preparation of this one. Thank you.
Thank you so much, sir. Now the session is open for discussion. Are there any questions someone wants to put up to sir? So if there are no further questions, thank you, sir.
No question, I just wanted to
Oh,
inform.
please.
Study come from an NGO sector. So if we require any residential training on is there any problem with the institute?
Yeah, definitely it will be there. So we used to provide our integrated farming system. I am working in integrated farming system. Now we used to provide some training. So if you wish, then you can come. We used to give training, including the agroforestry and all. We used to give.
Can I get your number?
And okay, I'll give you. Okay. Thank you.
So much, sir. So moving forward to the last lecture for today's first session, that will be delivered by Mr. Motiur Rahman, AFS DAO, SF Division, Karimganj. And sir will be joining us online. Sir, am I audible to you?
Am I audible, sir?
Yes, sir, you are audible. So can you kindly share your slides of PPT?
Yeah, ma'am. I'm sharing my slides.
So will be delivering lecture on department nurseries, potential and challenges. In the meantime, I now request our respected director, sir, to kindly felicitate Dr. Pravesh Devan, sir, with the presentation.
Is it visible, sir?
Yes, sir, it is presentation.
I am Motiur Rahman, AFS, Deputy Conservator of Forest and Divisional Forest Officer, Social Forestry Division, Karimganj. There is a big thank to Director Sir, RFRI, and Dr. Sonali Ghosh ma'am for allowing me to participate online in this workshop. I want to confine myself within the Department Nursery of Forest Department, Assam. I just starting my slides with the logo Vriksha Andolan 2023. It's moving. Yes, 1 minute. This is the introduction slides. It is nothing but history of the raising nurseries. Nursery divisions already several resource persons has described. During the past century, expansion of the urban had taken place and the forest resources were exploited to meet up the growing population in the world. That is why tree seedling production has become a fundamental tool for addressing afforestation, reforestation, restoration, and conservation of degraded forests. Nursery practices for tree seedling production have evolved considerably over the past several decades in accordance with the technological advances, increased and development and changing customer demands. To replace the invasive plant species with the indigenous plants, it has been felt to produce seedling of native species, which was the part of the rise of the environmental movement in late 20th century. Categories of forest nurseries. In our department, we are developing two types of nurseries based on the CAMPA norms. One is permanent nursery. It is established in a fixed place with full facilities, all facilities having areas of 1.5 hectare to 10 hectare to produce 2.1 lakh to 25 lakh seedlings in a year in Assam. Here, I want to mention one thing. This year, we are going to raise three numbers of big nurseries in three districts. One is in Gwalpara, another in Sunitpur, and the third one is in Karimganj, wherein 25 lakh seedlings will be produced in each nurseries. Later, coming to temporary nursery, it is nothing but field nursery where plantation is raised. This stock is used for the plantation to meet up the requirement of the plantation to be raised, and the area of the field nursery or temporary nursery is taken based on the requirement of number of seedlings, colonial propagules in the proposed plantation for vacancy filling over the maintenance period, which can be calculated by applying this formula, wherein age implies the area of the nursery in square meter, small a area of plantation to be planted in hectare, n small n number of seedlings required per hectare of plantation, and capital N number of seedlings per standard bed of nursery. Standard bed means 10 meter by 1 meter. Guidelines for raising nursery of to produce quality planting materials. Regarding quality planting materials, several research persons has described. I will just showing the slides to take some precaution measures to produce the quality planting materials. Workers of a nursery should receive professional training. In our department, we are having so many field staff who have been given training for raising nursery and other things in the field regarding plantations. Seed should be collected from designated well-maintained seed orchards. We are not having any designated seed orchard in our state, but we are having so many experimental plots and preservation plots from where we can collect quality seed. We are having 36 number of experiment and preservation plots from where we are collecting seed, and the trees of that project experimental plots and preservation plots are superior quality, which are being selected based on the morphological and phenological method by the field staff. Mature pods fruits should be collected just prior to falling the seeds, extracted early as possible without damage. Using fresh seed for seedling production is advocated. If seed has a long viability period, it can be stored to be used later. In case of vegetative propagules, fresh scion or buds should be collected from identified mother plants and used for grafting or budding with 12 pods. Seedling germination and grafting budding success percentage can be increased by following appropriate pre-sowing and growth regulator treatment of seeds and vegetative propagule respectively. Growth regulation treatment of seeds. Actually, from my field experience I acquired over the years, I can just recommend that every seed should be treated with water first. It may be hot water or cold water. This is the first principle I know. I have come to know from field experience. Here, where feasible, fruit spores should be collected directly from the tree to protect them from getting exposed to soil moisture or soil microorganisms. Seeds should be collected from several mother trees to ensure genetic diversity on the mother side. Also, collection of mature seed maximizes seed viability, seed storability, and result in good quality seedlings. In order to collect good quality seed, information on seed collection period is essential. A few MPITS produce seed every year, while some other species produces seed only in a cycle of multiple years. However, most species produces seed only during the certain period of the year. Collection of seeds, as I already told few words regarding seed collection. Here, four types of sources are available. Seed orchards. We are not, our department is not having any seed orchard. Seed production we are having. It is seed production areas apart from the preservation plot or experimental plot. Our field expert can collect seed from the plant trees available beyond the circle of the preservation plot and experimental plots. And the fourth one, population of seed trees. It is actually outside forest areas. It's currently found in the farmlands. Big trees with having clean bole and morphologically superior quality. From that trees also seed are collected. This is the seed collection procedure here mentioned. Seed collection directly can be collected from the small trees physically, and from the big trees we can collect seed before, as soon as it gets maturity. When the seed get mature, we can collect them by shaking the tree, which fall on polythene sheet or gunny bags laid under the trees. And other ways, there is another method by using ladder. By using ladder, we can collect the seed from the trees, plant trees. Nursery technique, as I have told already, that irrigation facility is the first tool, first parameters, first equipments, first provision facilities for raising a nursery from where quality planting material can be produced. Specific recommendations on the rooting medium, including mesh chambers, cold form frames, shade net, house, and induced rooting should be followed. Here, from my experience, I can express, as I told earlier, every seed of any species should be soaked in water, irrespective of the season. And then that seed should be dried in shade for 6 hours to 12 hours, and then to use in nursery beds or polypods. Here I got experience regarding the shade net and cold frames and mesh chambers. I used to use simply 50% agronets at the beginning of sowing of seeds in nursery beds or polypods. Here, there are two types of nursery beds. Nursery we are using. One is mother bed, where germination takes place, and other is naked bed, where naked seedling are produced. From mother beds, we transplant seedling to the pots. Here in our department, we are using 5" by 8" polypod bags, wherein we are transplanting the seedling from the mother beds. Mother beds, as already told, it is the germination bed only. From mother beds, we apart from the transplanting of seedling from mother bed, we can also sow seed directly in polypod. But here care should be taken that there may be some mortality in polypod, for which we simultaneously broadcast some seeds in mother beds, from where the seedlings are transplanted to the polypods, where directly sown seeds has failed to germinate. Rooting media should be used as per recommendations. We are recommending in our department, if a normal soil is used, then we the mixture of the rooting media is made of 1 part soil, 1 part sand, and 1 part cow dung or FOM. Here, in some districts like Dhemaji and Lakhimpur, where soil condition maximum sandy, where soil is not required. 50% that type of soil and 50% FOM is required to make the mixture. Here in Barakbali, I have seen the soil type is alluvium, totally cart soil. It is cart soil, locally known as cart soil, and we can say the alluvium, wherein maximum sand is required. Where we can use here 1:2:2. 1 is soil, 2 is sand, and 2 is cow dung or FOM. Regarding root shoot ratio, it should be maintained properly to produce plants free from weeds, disease, and pests. Standard phytosanitary measures should be followed. Application of optimum and balanced nutrients and use of biofertilizer should be encouraged. In case of fruits, if seedlings are being used as a root stock, homogeneity should be maintained by using clonal vegetative propagated plants. A minimum of 2 to 3 shifting of saplings and seedlings with root trimming in polybags, root trainers, to avoid root penetration in the bed is essential for raising healthy plants in nursery. Here in Assam, we are also following this system. 3 to 4 shifting is done in nursery seedling, mainly polypoded seedlings, wherein there are 2 holes in the side of the polypods, from where if seedlings are not shifted to other places, or inter shifting is not made, then this roots penetrates to the soil, below the soil. And that is why this process is being followed in our departmental nursery also. During lifting, transportation of seedlings, precautions are departmental experts are there. They are following all the precautions to save the seedlings. Species information which are kept in mind to kept in mind for quality information in nursery. To species wise, how to keep planting materials safe from damage. All are regarding all the matters of training are is given to the our staff involved. TPM provenances. Quality planting materials cover not only quality plantable seedlings or vegetative propagules, but also seeds collected from recognized seed orchard or plus trees. As I already told, our field staff, some of field staff are very expert to identify the plus trees outside the preservation plot or experimental plots, from where we direct them to collect seed for the nursery. As I already tell, there are 36 developmental preservation plot from where we collect seeds from plus trees available there. Potential and challenges of forest nurseries. Here, as logo in the first slide shows, the Brick Andolan 2023. We are going to plant 1 crore seedling this year, and we are hurriedly raising some nurseries for developmental seedlings. As notified, Assam outside tree outside forest rules, wherein 3 schedules have been schedule of species have been mentioned, where 70 numbers of plant species have been exempted from requirement of permission from forest department for felling and conversion for promotion of agroforestry nation. This year also, 2024, the Assam government has decided to plant 5 crore seedlings, for which our PCCF has directed to raise mega nurseries, that is 10 hectare each in 3 districts. One is Gwalpara, another is Sunitpur, and the third one is in Karimganj, to produce 25 lakh seedling each in 2024. Challenges we often faces in raising nurseries: lack of sufficient technical experts in the department of scientific nurseries in a large scale. We are having field expert, but this number is not sufficient. The lack of proper seed orchard for collecting quality seeds in the state. Seed orchard we need to establish. Biotic pressure due to growth of population, due to growth of large number of populations, we experience sometimes pressure on forest as well as in nurseries. Adverse weather due to climate change. Sometimes high rainfall occurs, sometimes low climate temperature affect the nursery seedlings. That is also a challenge. This is the notification tree outside forest sustainable management rule 2022, wherein I have told that there are 3 schedules containing 70 species. Certification. We though have records of the saint of custody of seed and or vegetative materials used for producing planting stock are being maintained by the accredited nursery at all stages, but proper certification is not being done due to lack of authorized body or institution in the state. Few days back, we learned from the CAFRI Jhansi that they are moving to state government to certify the quality planting material being produced departmentally in several states. I think RFI Jorhat may also take some step to notify such type of bodies by the government. Strategy, I think the government is taking to notify the such body. This is suggested parameters as suggested by CAFRI as well as by me also here. Some changes I am going to make based on my experience. These are the species wise some characteristics parameters of seed quality planting materials, seed collection period, period of nursery, age, height, collar diameter. Collar diameter here is the junction of stem and stem. That is, it is the ground level portion of seedlings. This is the dimensions which are suggested to be standard, and that will be quality planting materials. In case of Kodam, I think here age of Kodam 6 months. It has been put by me, but in the CAFRE assessment it was given mentioned as 12 months. Kodam 6 months old Kodam is sufficient, and Kodam generally from my field experience I can say 6 inch, that is 15 centimeter height Kodam is fit for quality planting materials to plant in the plantation site. Here in mention has been mentioned 30 to 45 centimeter, but it should be 15 centimeter to 30 centimeter. Similarly, eucalyptus, regarding eucalyptus, here in CAFRE data mentioned 3 months in nursery. 3 months old eucalyptus cannot be used in the plantation area. It should be at least 10 months. But rest are the, I am agree with this data. These are the few data of few species. This is the departmental nursery at Dukhipur under Korimganj Social Forestry Division. It is Central Album, Bagasandam. This is the uniform seedlings with good health. This is another nursery. It is Equileria melangensis. It is a departmental nursery, 1.5 hectare Campa nursery. The age of the seedling is 10 months. It has already been measured to plant to the plantation site, and these seedlings have been fixed for Briksha Andolan 2023, Amrit Briksha Andolan 2023. This is another nursery departmentally created in Korimganj Division of Headquarters. These Kodams are 5 months old. Authorized body or lastly, I just want that these are executed. Authorized body or institution need to be set up for accrediting, certifying quality planting materials in the state, and the government of Assam is taking strategy for the purpose. Awareness of promoting exempted species under TUF need to be created. I like to request all of the participants to create some awareness among the general masses, among the especially self-help groups, tea gardeners, etc., regarding the notification issued on TUF. Provisioning of finances for creation of large-scale developmental nursery in the state. In future, huge amount of finance will be required for creation of large number of developmental nursery in the state. Thank you.
Thank you so much, sir, for the presentation. We come to the conclusion of the first sessions for today, and we shall now begin with the valedictory session. We shall start with the certificate distribution.
Please, ma'am, repeat. Hello.
Hello, sir. Anything you wish to convey?
This audio is not good, the poor audio.
Anything you wish to convey to the forum?
Audio is not coming clear. Am I audible, sir, ma'am?
Sir, you're audible. Anything you wish to convey to us, sir? Am I audible to you?
Just I want to convey the RFI Jorhat to move for authorized body certifying the quality planting materials in the state.
Thank you so much, sir. We certainly look forward to implement as per your recommendation. Thank you so much for joining with us.
Thank you, sir. Thank you.
So we shall now start with the weekly session with the distribution of certificates to the participants. So may I now request our respected director, sir, to kindly felicitate Mr. Aliram Saikia with a certificate. I shall be calling the names one by one. If you can please collect your certificates from director, sir. This Mr. Vidyadhar Sonowal. Mr. Diganta Chetia. Mr. Vidyadhar Sonowal, and then Mr. Diganta Chetia. Mr. Simanta Gogoi. Mr. Diganta Chetia. Then Mr. Simanta Gogoi. Mr. Sunil Kumar Datta. Mr. Viju Datta. Mr. Ujjwal Thakur. Mr. Bhaktalal Upadhyay. Okay. Mr. Anjan Hindu. Mr. Bimal Gorgohain. Mr. Irfamul Haq. Mr. Arfan Hussain. Mr. Mridul Kumar Mehdi. Mr. Anup Jyoti Borgohain.